welcome to the Society for Immune Polymorphism Spring 2024 webinar. This webinar is on advances in long-range sequencing for the analysis of human leukocyte antigen genes. We've got two speakers today. I'm going to introduce the first speaker, who is Professor Alexander Dilthey. Professor Alexander Dilthey is Professor of Genomic Microbiology and Immunity at the Institute of Medical Microbiology and Hospital Hygiene, University of Düsseldorf. He obtained a bachelor's in computer science from University of Düsseldorf in 2008 and a doctor of philosophy in statistical genetics from the University of Oxford in 2012. His current areas of research include analysis of population genomics and the development of bioinformatics algorithms to enable these analyses. Today, Alexander will be describing his current work on long-read sequencing for HLA typing and an assembly of complete MHC haplotypes.
Well, first of all, hello. Thank you very much for the kind introduction, Martin, and I'm very happy to be here to have the opportunity to talk about long-read sequencing applications in the HLA and in the MHC. First of all, before I get going, let me very briefly say a few words on the kind of stuff that we're interested in, the kind of stuff that we're working on. So we're mainly bioinformatics lab, working in computational immunogenomics. We develop computational models for inference in complex genomic regions, develop methods for long reads, and also sequencing-based diagnostics. We're not purely bioinformatics. We also have a small sequencing facility, which helps if we want to generate and analyze our own data. Now, the title of that talk contains long-read sequencing. So first of all, what is long-read sequencing? So we currently have three dominant sequencing technologies: Illumina, PacBio, and Oxford Nanopore. Illumina is a short-read sequencing technology. These other two are long-read sequencing technologies. Short reads means typically on the order of a read length of 100 to 250 bases at a really, really low error rate. Long-read sequencing technologies, on the other hand, are able to sequence much longer fragments of DNA. So starting with perhaps 1,000 bases of DNA and going up to a few 100,000 bases. At a somewhat increased error rate, although one has to say that the error rate of these technologies has really been falling very steadily over the last years. And long-read sequencing technologies can also be used in targeted and whole genome applications. I believe that a talk by Oxford Nanopore is coming up at the next session, so I will not say anything about how these technologies actually work. Now, typically when I give a talk about analyzing the MHC, the first thing I do is I introduce the MHC, the major histocompatibility complex. But I think in that context here, that will likely not be necessary. However, allow me to add a little bit of context with respect to what follows. So the MHC is, of course, super important for transplantation in terms of HLA matching, but as most of you will know, it is also the region with the highest number of disease associations in the whole human genome, and many of these remain incompletely understood. So if I say incompletely understood, what do I mean by that? By that, I basically mean that the underlying causal allele that drives the association on a biological level has not been identified yet. And why is that? That is the case because the MHC is characterized by extensive linkage disequilibrium. That basically means statistical association between alleles at different loci. If you have extensive linkage disequilibrium, pinpointing the causal alleles that underlie an association really requires reliable sequence information across the whole region. So basically what you need is you need to be able to resolve the complete region on a base-by-base basis. And as it turns out, and as we'll see on the next slide, that's not so easy. And in addition to that, also an additional point, let me also point out that not all associations in the MHC are driven by differences in the HLA peptide binding regions of the HLA genes. For example, there are very well-known associations that are driven by regulatory SNPs, and also associations, for example, with the C4 gene in schizophrenia. So it's not all about peptide binding in the MHC. Now, we want complete MHC sequence information, but that is actually not that easy. And allow me to present two little pieces of evidence in support of that statement. So first of all, what we're looking here is a visualization of the alternative MHC haplotypes of the human reference genome. So the human reference genome contains, for a number of regions, and the MHC is one of them, contains alternative haplotypes to basically model and characterize the genomic complexity of these regions. And what you're seeing here is now basically these MHC haplotypes, and whenever a region here is highlighted in red, then that means that the underlying sequence is still undefined. So even in the human reference genome, the MHC haplotypes are not fully complete. Additional bit of evidence. Now you may say, okay, what do I care about the human reference genome? Could just, for example, do whole genome Illumina sequencing and then do variant calling in the MHC, and that will give me a complete set of variants that are present in that region. What you see on the right-hand side here are some aligned short-read Illumina sequencing reads from a clinical sample. And the IGV visualization here is focused around the MHC class 2 region and HLA DRB5. What you see here, if you know a little bit about analysis of short-read sequencing data, you see these patterns of strongly fluctuating coverage. That's strongly indicative of the fact that the alignment in that region here really didn't work very well at all. And any kind of variant calling that is based on alignments of that quality is bound to be highly unreliable. So even if you do standard whole genome sequencing, you're not going to get particularly reliable sequence information across the complete MHC. And that, in turn, then complicates the disease mapping use case that we have in mind. Now, why is analyzing the MHC so complicated? Three factors. First factor is the allelic polymorphism of the HLA genes, which makes it complicated to correctly map reads to the genes that they belong to. Then secondly, the structure variation, for example, as seen here in the class 2 region. And then last but not least, that allelic polymorphism point is basically also there's an interaction with the fact that there are also sequence homologies between the genes. So not only are the genes within themselves very polymorphic, different alleles at different genes can also be highly similar, which adds another layer of complexity to the read mapping problem. And for example, if you're interested in the extent of a sequence homology between different genes, we quantified that in a 2016 publication. Now, where can we go from here? We have basically two alternative approaches that we may pursue. We could become more intelligent about the analysis of the generated read data, or we could try to use better and more informative data. And of course, in this talk, I'll briefly touch upon both points. In terms of becoming more intelligent about the analysis of the read data, I'll briefly talk an approach called population reference graphs that you can use, for example, to do HLA typing from standard whole genome sequencing data, be these long or short reads. And then in the second part of the talk, I'll talk about the use of long-read sequencing data. And the intuition here is, in the context of what I just said, that these longer reads can be more confidently mapped and more confidently aligned to the genomic locus that they originate from. And using long reads, we're now in a position that you can use these to infer complete MHC haplotypes. And of course, it may also be possible to combine both approaches. Now onto the population reference graph. So a population reference graph is a mathematical model of a population of genomes, and I'll show an example in a bit. But basically, the way to think about this is that you can take the reference genome and substitute it with a population reference graph that models the variability in your population. If you want to construct a population reference graph, what you start off with is a reference panel of resolved sequences, shown here on the left-hand side. And you then use a graph construction algorithm that typically identifies and collapses regions of homology. And these algorithms then build data structures that basically look like this here. That's a graph because it consists of nodes and edges. The edges connect the nodes. And you can see that all the input sequences here on the left-hand side can basically recapitulate it as a path through that graph model. But in addition to that, you can also generate novel sequences that were not part of your reference panel. That is basically the way that that mathematical model here incorporates the effect of recombination that acts on human haplotypes. There are a number of research questions around these graphs. So, for example, how to build them and how to use them most effectively. Graphs are a hot and very relevant topic right now in human genomics in general, in terms of, for example, pangenomics. But it is worth pointing out that it was basically the field of immunogenetics with the highly variable haplotype structures that these models were first developed and explored and actually validated. So you can use these graph-based approaches to improve analysis of MHC sequences. One challenge here is that we need to integrate, as a first step, all the available information on HLA sequence variability. We need special algorithms for that, which we've developed. And if you apply these algorithms, you then get these beautiful graph-like structures here, for example, representing one exon of HLA DRB1. And using tools, graph-based tools, you can then infer, for example, the genotypes of the classical HLA alleles with very high accuracy. What I'm showing you here is validation results for a tool called HLA-LA that was developed in my lab. And if you use long-read sequencing data here, for example, from a very diverse African cohort, you see that you get very, very, very high accuracy HLA types. So that's great. If you want to analyze long reads, and if you want to do HLA genotyping, for example, HLA-LA is a tool that enables you to do just that. Now, on to the second part of the talk, long read sequencing specifically in the MHC. So in the introduction, we already introduced these two technologies, the two relevant long read technologies, PacBio and Oxford Nanopore. And so you may wonder, how may these help specifically in the MHC? And to think about that question, or to approach that question, let's look at the structurally variable regions in the MHC. So what have we got here? We have the MHC class 2 region that has a length of between 80 and 250 kilobytes per haplotype. With the C4 region on the order of less than 100 kilobases, and we have structure variants in the class 1 region, on the order of 55, 60 kilobases. The important thing is that with these long read sequencing technologies, we are now able to generate reads that are as long as or comparable in length to the length of these regions. If you know a little bit of genome assembly, you know that the important bit is that basically once you have a read that spans your complex region, then your assembly algorithm is typically able to confidently resolve that region. So using these long read sequencing technologies, we may actually be able to infer complete MHC haplotypes, at least in theory. And we wanted to test that theory. And so in a collaboration with Paul Norman's group, we set out as a first step to completely resolve the MHC reference haplotypes of the human reference genome. You remember earlier I said that the human reference genome contains 8 alternative MHC haplotypes. 6 of these are incomplete in the sense that they contain unresolved sequences. So we thought, okay, let us try to resolve these. A little bit of background on these alternative haplotypes. So they come from 8 MHC homozygous cell lines that have already been characterized as part of the MHC haplotype project. The cell lines were selected because they carry haplotypes that are important in terms of autoimmune disease susceptibility, or because they have high population frequencies in European populations. And they also comprise all DR groups apart from DR1 and DR8. So they're not fully representative, but, well, they're first step towards capturing relevant diversity in that region. And here I already showed you that visualization here of the 8 haplotypes in there, 6 are incomplete, 2 have already been resolved. Okay, so first thing we did was we obtained the original cell lines for 5 of the 6 incomplete haplotypes. The 6th cell line we couldn't basically get hold of yet, and we also included another cell line, Cas116, because that contained another DR structure. So what did we do? We combined 4 different sequencing approaches and applied that to the 6 cell lines that I just mentioned. And these sequencing approaches included Oxford Nanopore sequencing, using the ultra-long protocol, where you have maximum read length basically over 1 megabase. We also carried out PacBio Hi-Fi sequencing, Illumina whole genome sequencing, and we also rolled in some Illumina targeted sequencing data that Paul and his group had produced earlier. The way to think about these different sequencing technologies is that the nanopore ultra-long whole genome sequencing gives you the correct structure. Then the PacBio Hi-Fi also helps with resolving the assembly and also helps with the base pair accuracy. The Illumina whole genome sequencing then is basically for the polishing, and the Illumina targeted sequencing data can then also further add to the polish. That little table here gives you an idea of the data that we generated. So for most cell lines, we generated on the order of 20 to 30x nanopore whole genome sequencing coverage, on the order of 15 to 20x Illumina coverage, and approximately 10x PacBio sequencing coverage. Now, of course, a recipe includes not only the cooking ingredients, but also how you analyze and combine the ingredients. That's the bioinformatics part. So we developed a bespoke recipe for assembling these into contiguous haplotypes. So first of all, we extracted MHC mapping reads by alignment against the existing incomplete haplotypes. Then we experimented with the two algorithms Canoe and Fly, and basically empirically varied the parameters until we had contiguous MHC assemblies. So basically the MHC being represented in one contig. And then we trimmed these contigs to the canonical MHC coordinates as defined by the PGF haplotype, and carried out multiple rounds of a process that is called polishing. And polishing basically means that you start with an existing draft contig and try to improve the base pair accuracy of that contig over multiple iterations. And so what did we do? So first of all, we used the nanopore data and called small variants with Medaka, integrated these into the assembly. Then we rolled in the Illumina whole genome sequencing data, called variants using GATK, also integrated these into the assembly. Then we integrated the targeted Illumina data that had been generated by Paul. And then after each round of polishing, we basically carried out the step of manual curation and inspection. During that step, we decided whether we would want to do another iteration. And at the very end, we had also a final round of polishing where we then integrated the PacBio Hi-Fi data. So that was our assembly recipe, which enabled us to go from these different 4 heterogeneous data sources into completely resolved haplotypes. Now, and that was great because that confirmed the initial hypothesis that we set out to test, whether we are now in a position to assemble complete MHC haplotypes if we bring in these long-read sequencing technologies. So answer to that is yes, it is now possible. Second step was, of course, to try to look into the variants found on these haplotypes. And that table here gives you an idea of the variants that we found. The different lines and rows in the table here represent the haplotypes. These 6 haplotypes here are the haplotypes that we generated. Cox is one of the 2 complete haplotypes that are already part of the human reference genome. But if we count variants, we count them against the PGF haplotype, which is the other complete reference haplotype of the human reference genome, and which is also the canonical chromosome 6 sequence. Now to briefly walk you through the table, the number of gene calls across these haplotypes was largely consistent. We also carried out C4 genotype annotation and annotation of transcripts, also no big surprises here. And you see that the number of SNPs relative to PGF varied between approximately 12,000 and approximately 14,000. The number of indels varied between approximately 2,000 and approximately 2,000, 400, 500. And we also had a few dozen structure variants per haplotype. One very important thing to note is that if you look at the variant statistics of Cox, which was generated previously using a completely different set of technologies, you see that the variant statistics of Cox really fall in the middle of the distribution spanned by our haplotypes, which indicates that the haplotypes that we generated are really reference grade quality. Otherwise, we would see, for example, an increased rate of SNPs that would be driven by sequencing errors when we compare to Cox. And we can also visualize the distribution of these variants. On the x-axis here, you have the MHC coordinate. On the y-axis, the different count of variants for different variant categories. And you see that these peaks here recapitulate the known areas of MHC hyperpolymorphism, for example, the class 1 and the class 2 region. So what can we use this data for? Well, I started off motivating the case for resolving complete MHC haplotypes in terms of disease association studies. We can now use these variant catalogs here and test the variants contained therein for association with disease. So that's already great. That really improves our ability to carry out fine mapping of associations in the MHC. And in addition to that, these fully resolved sequences also enable us to address interesting questions about MHC evolution. For example, based on these fully resolved sequences, you can now really carry out a high-resolution analysis of MHC class 2 sequence homologies. What you see here is a visualization of the class 2 regions of the haplotypes that we analyzed. And basically, these different colored blocks here indicate regions of homology. So you see that you start with a region of homology here on the left-hand side towards the DRA gene. Towards the right-hand side, you also have homologous sequences with the DRB1 gene that's also universally present. But then in the middle here, you see that these colorful patterns really indicate that really give you an idea of the different sequence fragments that were shuffled over the course of MHC evolution. And the fact that basically a lot of sequence shuffling had occurred over evolutionary timescales for the MHC had, of course, been well known beforehand. But now with these complete sequence resolutions, you can now characterize them at high resolution. And in addition to that, we also confirmed that there are no additional previously unknown structural variants in that region here, which was also an important finding. Another thing that you can do is you can carry out a fine-grained analysis of repeat elements. Again, that plot here shows you the MHC class 2 region, and each of these panels here shows you the distribution of repeat elements for the different haplotypes that we analyzed. So within each panel here, you have basically one little line for each haplotype. And whenever the repeat element is present, well, that indicates that at that position in the haplotype, that particular repeat element, that line 1 element is present. So for example, to give an example here, that block here stands for the line L1 repeat element. These black dots here indicate that at that homologous position, that repeat element was inserted across all of the analyzed haplotypes. And that analysis that we carried out based on these complete sequences confirmed that the class 2 sequences were heavily affected by repeat element insertions. These have been previously implicated in non-homologous recombination between the different haplotype structures in the MHC class 2. And so based on that, these sequences we can now really carry out a fine-grained analysis of the evolutionary history of that region. We can try to better understand where the different MHC haplotype classes how they emerged, how they're related, for example, to primate haplotypes, and also which evolutionary forces were behind the patterns that we now observe. And to basically finish off that section, let me show you a plot of the visualization of sequence homologies across the assemblies that were generated. I will not make any further comments on that plot here, apart from the fact that I think it should be immediately obvious that you really have a region of that is highly complex, and that the intricate patterns of that region, it's really great that we can now study them in detail. So conclusions and perspectives. Long-read sequencing enables multiple applications in the MHC and HLA. So first of all, you can use long reads for HLA typing, for example, using the graph-based approaches that I talked about earlier. Long reads also enable you to completely characterize whole MHC haplotypes. Doing that, however, is still not entirely routine. It is still a bit challenging in terms of the required input data, and also a bit challenging in terms of the bioinformatics, as was probably evident from the description of the bioinformatics pipeline that we used for assembly. Do you need to combine 3 different sequencing technologies in the way that we did? No, there are also alternatives. For example, the Human Pangenome Reference Consortium also has an assembly recipe that enables resolution of high-quality MHC haplotypes from whole genome sequencing data. In their case, they used high-coverage PacBio data and combined that with TRIA information. So that, for example, is an alternative that can also work. What can we do with these completely resolved haplotypes? We can use them as a basis for fine mapping of disease associations. But in order to do that, we need to go one step further. We need to basically go from set small number of completely resolved haplotypes to population scale genotyping of MHC variation. So how could that work? And that is basically the third element, the combination of graphs and long-read sequencing that I alluded to towards the beginning of that talk. So the program for basically achieving that goal here works as follows. So we use single molecule long-read sequencing technology to generate additional haplotypes. And for example, we are involved in a collaboration with Paul Norman's group to do exactly that for a few hundred of additional haplotypes. We can then use these completely resolved sequences to build improved genome graphs. And based on these genome graphs, we can then do cohort genotyping in the largest human cohorts that we have, such as all of us or the UK Biobank. Then at that stage here, should finally be in a position to carry out a comprehensive analysis of what is driving the associations in that single most important region for human disease genetics. And with that, I would like to thank you again for the invitation. I would like to thank the people who contributed to the data that are generated. In particular, I said that already, but that's really a great collaboration with Paul Norman groups. People who in particular deserve to be pointed out are Torsten and Stefan from my group, Nick and Billy from Torsten's group, and of course, Libby from Stanford. And if you're interested in the software that I mentioned, everything that we do is open source. You can check out our GitHub. And last but not least, I would like to thank our funders, including the German Research Foundation, the NIH, and also the Mancho Foundation. And you, of course, for your attention. Thank you very much.
Fantastic. Thanks, Alex. I think we have time for a question. It's 1 minute before the half-hour point. If anyone has one, they can raise their hand or post it in the chat. I was going to ask you about the human pangenome reference. So are you saying that like that data as it's now available on, I think, something like 47 individuals, can you just use that to augment the graph and it sort of gets better as you add more reference haplotypes to it?
Yes, precisely. So important point one is that the MHC haplotype quality of these pangenomic projects is really amazing. And then secondly, yes, the general idea is to keep basically adding to the graph until you've represented all relevant variants. And what relevant means may depend on your population and also depend on your, for example, target minimum allele frequency.
Yeah, I understood that there was, they're, I believe, using some of the same samples as HapMap. So there's a goal of getting a lot of sampling of diverse populations there, which is great. What was the short, you mentioned the range of the class 2 region and the low end seemed especially low. Is that like some anomaly or is that just the DR4 versus the rest? I mean,
yeah.
the length.
It's basically the latter. It's the shortest basically class 2 haplotype structure that does not carry any functional DRB genes apart from DRB1. I think it's actually DR8. I keep on messing up DR1 and DR8, but I think it's DR8.
DR8 I understood was sort of a deletion where the DR is a fusion of the DRB3 and the DRB1. Well, fantastic stuff. I think we should break here because we have a second speaker and I think you have to go to another meeting, but thanks a lot for your presentation. And now I'll hand it back to Nima to introduce our second speaker. Thanks, Alex.
Thanks, Martin. So it's my pleasure to welcome Dr. Dianne De Santis to give the next presentation. Dianne is a clinical scientist in the Department of Clinical Immunology at Pathwest Fiona Stanley Hospital in Perth, Australia. She completed a PhD investigating the role of KIR receptors in hematopoietic stem cell transplantation. She's responsible for the stem cell transplant patient search program in Western Australia and for developing new technologies to improve HLA typing methods used in both solid organ transplant and stem cell transplant as well. Dianne and her team were the first to publish on the novel high-resolution HLA typing method utilizing single molecule sequencing on the Oxford Nanopore platform for deceased organ donor allocation. These methods have recently been implemented for high-throughput stem cell transplant work on clinical samples and registry typing. Dianne is the Vice President of the Asia Pacific Histocompatibility and Immunogenetics Association and a counsellor on the International Histocompatibility Workshop Committee. Today, she'll be giving us some information on her work on revolutionizing high-resolution HLA genotyping for transplantation assessment, the validation, implementation, and challenges of Oxford Nanopore Technologies Q20 plus sequencing. Welcome, Dianne. Thanks a lot.
Thank you, Nima. Thank you for the warm welcome, and thank you for the invitation to share what we've been doing in Perth. So I hope you can see my slides. Can see my slides,
we can
Nima?
see those.
Yeah. Okay, great. Yeah, so just a disclaimer. So Oxford Nanopore technology products are not intended for use for health assessment or diagnosis, treat, mitigate, cure, or prevent any disease or condition. And any of the work that I'm presenting today has been done in-house without the collaboration of Oxford Nanopore. So it's all in-house development. So I thought I'd just start with a little bit about our journey over the decades and how our lab got to where we are today. So in 2010 or 11, we started looking at the short read sequencing or next generation sequencing for HLA genotyping. And that was just to overcome some of the issues that we already know existed with Sanger sequencing. And we were the first, or I was the first to introduce an NGS workshop at the 13th International Workshop. And then we worked for a few years towards the implementation of iron tolerant sequencing for clinical and registry typing in 2014. Around 2016, Oxford Nanopore started releasing some of their early access products, and we started looking at the long read chemistry with Oxford Nanopore. We did look at PacBio as well, but we decided that we would continue on the Oxford Nanopore pathway, mainly due to the fact that the capital was really low to invest in the equipment. In May 2020, we were the first to implement Oxford Nanopore technology for rapid 2-field typing resolution for deceased donor organ allocation. So high-resolution typing in about 4 to 5 hours. And then over the next few years, we started looking at the R10 flow cells for clinical samples and registry typing. And in 2023, we implemented R10.4.1 for all our HLA typing requests. And recently this year, we further improved our rapid typing method for our deceased donor organ program to just under 3 hours. So we've been on the Oxford Nanopore sort of journey for the last 8 years. And so just a bit about sequencing with MinION technology and how it works. So shown here is the double-stranded DNA, which is ligated with an adapter. The nanopore here is shown in blue. So it's a protein nanopore that sits in an electrically resistant polymer membrane. We have these tethers that sit in the flow cell and this enzyme protein motor. And so how it works is the double-stranded DNA, there's an adapter that's ligated to the DNA strand, and that's bound to this enzyme motor protein. And this protein essentially unzips the double-stranded DNA and pulls out through the nanopore. And as it moves through the nanopore, as the DNA moves through the nanopore, each nucleotide disrupts the pore in a has a unique electrical signal which is recorded and then converted into the base. And this is can all be done in real time. So really, as the previous speaker spoke about, the increase in accuracy has improved with the development of new flow cells and chemistry. And so in 2022, Oxford Nanopore released the R10.4.1 flow cell and Kit 14 chemistry. And really, the development was with a pore that had a longer pore, which was claimed to be better at sequencing the homopolymer regions, improved accuracy, faster speeds, reduced errors, and a new motor, E8.2, again, improving the accuracy and more consistent movement of the DNA through the pore. And this also allowed for duplex sequencing, so sequencing in the forward and reverse direction, again, allowing for higher accuracy. In terms of the MinION devices, the MinION device, the MK1B is 100 grams in weight. It is run through a USB 3 cable. You can plug it into a laptop. It's portable. The MinION flow cells, the flow cells have 512 channels. Each channel has 4 wells with a total of 2,048 membrane wells, and each well consists of 1 nanopore embedded in through the polymer membrane. Sequencing speed is around 450 base pairs per second, and it can be reused for a total runtime of 48 hours. We also have the single flow cell, fungal flow cells, which have 126 nanopores. They're single use and generate up to about 2.8 gigabases of data. They also have a high throughput benchtop device, the GridION. This is a self-contained, easily deployable benchtop device designed to run and analyze up to 5 independently addressable MinION or fungal flow cells. It theoretically can output 50 gigabytes of data per flow cell. And as I said, it holds 5 flow cells, so up to 250 gigabytes of data. So for us, that's around 160 samples per day. It's a small footprint. There is no capital cost, so you pay for the consumables and you acquire the instrument. And it does allow for real-time base calling for any urgent samples or for disease genotyping or any urgent clinical samples. And we did experience some issues within our health department firewall. So there were firewall issues where we couldn't access the ONT URLs for troubleshooting and updating software. So initially we ran this instrument offline until we were able to change the firewall rules through our health department so that we can run the GridION online. So that was a little bit of a challenge working within a health network. So why MinION sequencing in the HLA laboratory? So when we started looking at long-range sequencing, there was a lot of people who were already doing next-generation sequencing, and so the number of HLA alleles discovered had exploded, and this made genotyping complex again. So we now have more than 38,000 alleles in the database, and we found that although we had moved to second-generation sequencing and we had reduced the number of ambiguities we were seeing with Sanger, we started to see ambiguities again with the short sequencing chemistry just because of the numbers of HLA alleles that were being discovered. Again, in solid organ transplant, at that time, around 2019, the low-resolution typing methods really did not equal the single antigen bead panel, or didn't represent the alleles on the single antigen bead panel. And therefore, we were excluding patients unnecessarily from organ allocation lists and transplants because we couldn't confirm allelic antibodies present in patients. And throughout the last decade or so, and prior to that, there's been an increasing in data published on the impact of regions other than the antigen recognition domains of the class 1 and 2 genes. And so there's evidence now showing that there are other regions that could be important in transplantation outcome. And these are just some of the ones that I've listed here. So obviously in the 1990s in the Perth lab, we talked about MHC block matching, and we showed that that was associated with patient outcomes. So that looked at matching across the MHC, not within just the class 1 and 2 genes. Nima Mayer in 2019 published some data looking at the impact of ultra-high resolution HLA matching in transplantation outcome, again looking outside of those antigen recognition sites. More recently, HLA-B leader matching in transplantation on survival, where exon 1 is important, and looking at DQ alpha and DQ beta 1 heterodimers in hematopoietic stem cell transplantation. So we want to start looking at other regions of the MHC in disease. So we asked the question, is MinION sequencing feasible for HLA genotyping? So we know it was long-read single-molecule sequencing, so we're able to sequence full gene, and this allowed us to phase across the gene and therefore reduce genotyping ambiguity, and maybe that led to better donor matching. We could sequence intron polymorphisms that could be markers for haplotypes, SNPs in regulatory regions which could affect expression, and other MHC genes that could be important in transplantation. This technology was not heavily dependent on bioinformatics, so there were no mapping algorithms required, just alignment, no need for expensive equipment, and the library preparation and sequencing was quite simple and therefore reduced the turnaround times for HLA typing. So at that time, there really wasn't any commercial assay that really that amplified the full gene. So we developed our own in-house targeted amplicon PCR assay. So we amplify the full gene for class 1 and DQ alpha and some of the class 2 genes. Well, actually, they're all full gene. And recently, we've added ABO into our class 1 reaction mix. So it's 4 reactions per sample. We pull the amplicons from each of the 4 reactions. We purify the amplicon, and then we perform the library preparation as per the ONT kit's recommendations. We found at the time that we were generating quite a lot of data, and analyzing that data using Linux commands to do the base calling and the filtering, etc., was taking up to 3 days. So we needed to develop something that used GPU power and that it was quick. And so we worked with Microsoft and BizData, who was a consultant in New Zealand, in developing a pipeline that is housed in the Azure environment. And so this pipeline takes the pod files from our network drives. We have a Loom agent that detects those files. Once those files are detected, they are uploaded into this Azure environment. They are stored there. And then this Loom agent kicks off the analysis once it has all the files that it needs, and runs the analysis through containers that have the base calling software, so Dorado software, nanofilter, so some filtering tools to filter on quality and size, and some QC tools to calculate sequencing metrics. That's all run through GPU and CPU virtual machines. Once that analysis is performed, it's sent to storage, but then also downloaded to network drives, where we then analyze the FASTQ files with NGS Engine. That has been published, so you can read more about that pipeline in that publication. So to validate this method for routine use, we started with a phase 1 validation, and this included analyzing or testing a panel of 42 samples that represented the major allele groups. We assessed the data for sequence quality and accuracy, and we did compare against our R10.3 and R10.4 data. We then performed a phase 2 validation or implementation review once we implemented the assay, and we assessed failure rates and reviewed quality metrics. We set some acceptance criteria for the development and implementation of the MinION sequencing. So with just over the years of our development, we understood that it was important that we had greater than 300 total map reads at greater than 80% mappability per sample, and that at least we had at least 20 times coverage per allele, so 20 reads per allele. All assigned barcodes should be detected. The sequencing failure rate was around 4%, so 1 in every 24 samples. Our sequence quality had to be greater than Q7, and sequencing accuracy and systematic sequencing errors, such as homopolymer and inversion errors that we were seeing with previous chemistries and flow cells, had to be improved, or should be improved with the new chemistry or comparable to the results obtained with the previous version. And of course, all genotyping results had to be concordant with known or previous genotyping results to the 3rd to 3 fields of HLA typing resolution. So phase 1 results, so we sequenced 42 samples. We obtained about 5 gigabases of data, which equated to about 750,000 reads. 93% of our reads were greater than Q7, with an average of around 8,600 reads per sample. And we had a minimum of 20 reads per allele for 736 out of the 737 alleles. We looked at read quality. So shown here in the top graph is mean read quality and median read quality for our R10.3 and R10.4 flow cells with the 109 kit chemistry, and then R10.4.1 with 114 chemistry. And you can see that there is an improvement in read quality from the 10.3 to the 10.4 and then 10.4.1. We also looked at raw read accuracy using BLAST and LASTL. And we did that for the 10.3, the 10.4, 10.4.1, and compared that to our ion torrent accuracy. And you can see that the raw read accuracy using both methods improved with the 10.4 series flow cells and chemistry. So around 98%, which was about 1% difference to the ion torrent accuracy. But when we did consensus read accuracy, we really got high accurate results or accuracy, and really comparable to our current 2nd generation ion torrent sequencing. So at 99.98% consensus read accuracy. We did see improved noise and signal to noise ratio. So in the top graph here is our maximum noise percentage per locus. So in red shows the 10.3 series flow cells, and you can see that the noise per locus was much higher than in the 10.4 series shown in blue. And again here, the signal to noise per locus ratio was improved with the 10.4 flow cell series. As claimed by Oxford Nanopore, there was a reduction in the homopolymer errors. So here we see in 9.4 flow cells, we see the inability to really call the bases around the homopolymer regions. We saw an improvement with the R10.3 flow cell, and then with 10.4.1, we really had no issues assigning bases around the homopolymer regions. And that was really due to the improvement in length of the pore and the ability to sequence through those homopolymer regions. Long read chemistry has the ability to phase exons across the gene and resolve the genotyping ambiguity that we see with short read chemistry. So in the top graph view, we see our short read chemistry, so our ion torrent sequencing for A2301 and 2402. You can see that the polymorphisms in exon 3 can't be phased with that in exon 5, but we have no issues doing that with the long read chemistry. In our lab, we started sequencing our negative control, and we started to see a little bit of barcode contamination, or what's known as barcode bleeding. And this is the result of incomplete inactivation of the ligase during the barcoding step, or bioinformatic chimeric reads. So shown here in blue is our negative control mapped reads with Guppy 6.5.7 base caller. And then when we switched to the Dorado base caller, we basically got rid of those chimeric reads. We didn't see those chimeric reads to the same proportion as we did with our previous base caller. So these contaminating reads that we saw in our negative control were really due to these bioinformatic chimeric reads, which occur due to the fact that when one read or one strand fragment of DNA that has a say barcode 1 moves through the pore, a second fragment of DNA with a different barcode moves through that pore quickly, and the bioinformatics can't tell between the two different strands of DNA. So it pieces them together as one strand of DNA. But we wanted to make sure that that was the only contamination that we were seeing. We do reuse our flow cells, and we do reuse our flow cells for our deceased donor typing. So we wanted to make sure that we weren't seeing any sample contamination between those uses. So what we did is we took 6 donors and we barcoded them with different barcodes. We knew what the HLA typing were for each of those samples, and we sequenced sample 1, and we washed the flow cell, and then we sequenced sample 2, we washed the flow cell again, and so on, and we did that for 6 samples. And then we looked for reads from the previous sample, and we did that for all the 6 samples. And we really didn't see any contaminating reads. So the highest percentage was 0.16%. Our background sits around 3 to 5%. So this was really well below the background of the assay. So there was really no issues with sample contamination after reusing the flow cells. In terms of our implementation data, since implementation in July 2023, we've run 111 runs. 2 of these sequencing runs did have lab bench contamination. We had new operators, and so we had some issues with contamination. So working cleanly and changing gloves and when working with the barcodes is really important. We had 59 sequencing runs without BSA in the priming solution of the flow cell. 57 of those were first-use flow cells and 2 were second-use flow cells. So shown here are the sequencing metrics we use to evaluate a sequencing run. So we always perform a flow cell check to determine how many pores are available for sequencing. Sequencing time, the number of reads generated in that time, the data yield, the median quality, read length, and then the percentage of reads that are greater than Q7 and those that are greater than Q15. And then for our internal positive control, the HLA mappability. And so you can see we get really good data for our first-use flow cells. But with our second-use flow cells, we really only get, we got half the amount of data. The quality was much less. And so we stopped using the flow cells for a second time. When we introduced bovine serum albumin into the priming flow cell buffer, we actually saw an improvement with our second-use flow cells. So shown here, we got good pore recovery. Sequencing was longer with the second use, but obviously we're sequencing with less pores, so that's proportional. We did obtain similar amounts of data off the second use flow cells. Again, the same quality and same mappability. So the introduction of the BSA really did help with stabilizing the nanopores for second use. And so since implementation, we also had a change in the base calling software, so from Guppy to Dorado. And so again, once you change your base calling software, you need to review your quality metrics because things change. So you can see our flow cell check, the data yield. So we collect about 6.5 gigabases of data per run for 32 samples. about 14 hours of sequencing. And with the new Dorado base calling software, we saw an improvement in read quality. Read lengths were similar and percentage reads greater than Q15. So we saw actually higher quality data with the new Dorado base calling software. And that really was probably due to the ability to do duplex sequencing with this software. Our sequencing failure rate was high initially in our validation, and so we put that down to essentially not having enough data. So we increased the amount of data per sample, and once we did that, we didn't have any sequencing failures. And again, incorporating the bovine serum albumin into the priming solution helps stabilize our nanopores for our second use flow cells, and probably in our first use flow cells as well. Following implementation and changes to those changes to our workflow, in 2,426 samples, the total failure rate was assessed to be 6.3%. 1.2% of those was attributed to poor sample quality, 0.57% was attributed to sequencing failures, and 4.5% were attributed to PCR failures. And just quickly, this was the development of our two-field rapid high-resolution typing method for deceased donor allocation. So in 2019, we started this development. In 2020, we published this data and implemented it for deceased donor typing. In 2023, we investigated improving the turnaround time of that assay by introducing the new Oxford Nanopore Rapid Library Preparation Kit. So essentially, DNA extraction remained the same, so 20 minutes for DNA extraction. We used a modified version of the all-type PCR reagents. So we modified essentially the thermocycling conditions to make that program shorter. We did an ample amplification, a library prep of 10 minutes, and use the MinION flow cells to do sequencing. And analysis takes about 5 minutes. So this rapid kit has this transposon complex, which cleaves the DNA and adds the adapters, and then attaches the sequencing adapters all in 10 minutes, compared to about an hour in the standard ligation kit 114. So no barcoding. We use the MinION flow cell up to 5 times. And now these types of methods are available commercially through Omexon and Gendex. So in conclusion, MinION sequencing is feasible for HLA genotyping. Improvements in sequencing chemistry, nanopores, and base calling algorithms over almost a decade have improved the accuracy of the technology and really has become equal to second-generation sequencing. Rapid high-resolution HLA typing for deceased donor organ allocation can be achieved in just under 3 hours using the rapid sequencing chemistry, and that's novel and world first. I'm not sure if anyone else is doing this. We see no sample contamination when we reuse our flow cells, and we can use the flow cells without barcoding if required. Sequencing a negative control does identify the presence of barcode bleeding or bioinformatic chimeric reads, so that's something just to keep note. And we did see a reduction in those reads with the new Dorado software. But these contaminating reads or chimeric reads are really in very low proportions compared to your real sequences. Incorporation of BSA in the primary solution has increased the stability of the nanopores and has allowed for the reuse of flow cells. And we've seen improvements in base calling algorithms, which has increased the accuracy of the technology. And long read chemistry has improved HLA genotyping resolution and allowed for the development of other MHC gene assays. We have a KIA project in our lab and non-classical HLA genes. That's a master's project. And as discussed in our previous talk, the possibility of using this technology to characterize MHC haplotypes. So just to finish off, just acknowledge the people in my lab. So NASA and Lynn, who did most of the early work in the MinION sequencing, Oxford Nanopore, who supported us, BizData and Microsoft, who helped us with the development of the pipeline. And that's all.
Brilliant. Thank you so much, Dianne. That was brilliant. As we are on the hour, we've got a couple of questions in the chat that maybe we can just go over quickly. The audience would like to know, in your experience, what is the limitation of the number of homopolymer nucleotides that Oxford Nanopore can call with high accuracy?
I would say probably, well, I mean, I haven't looked at it specifically, but 7 to maybe up to 9, 7 to 9.
Okay. And are you seeing any sort of bias to one nucleotide over the other, which is something we've seen sometimes in our data?
We don't see that in our 10.4.1 data. We did see that in our 10.3 data.
Right.
Do you mean like an inversion? We called it inversion error, where we seem to have a G introduced when it should have been an A, for example. Is that what you mean?
Yeah. Or just in homopolymer regions in general, that you do tend to see one nucleotide possibly being a bit more problematic, I guess, than others.
No, we don't see that in our 10.4.1. And I will say that we're using NGS engine, and so it's not well, as far as I understand, we're not using the new algorithm yet. It's not cleaning up any noise or anything around the homopolymer, and I know there are some softwares that do that, and so they don't see any homopolymer errors. But we haven't really had any issues with homopolymers that are within exons. We've been able to call them. The DRB1 genes, we can't analyze the introns of DRB1 or DRB genes with all the repetitive regions. So we do have to exclude the intron 1 of DRB genes. But that's really the limitation still.
Yeah. Okay.
Yeah.
We have one more question. If the fraction of sequence reads are not aligned against a known allele, which may be seen in cancer, how are these sequences dumped? How can such sequences be further salvaged and analyzed at a subclonal level?
Sequences are not aligned against a known allele. Are these dumped? I'm not sure.
I guess that's along the lines of, have you got any experience, particularly in sort of malignant disease, in stem cell transplant patients? Occasionally, I think you can see novel alleles that are derived from the cancer itself. I wonder whether it's easy to kind of separate that out from the germline HLA types. Have you got any experience of that with Oxford Nanopore?
No, I'd say I haven't got any experience in that. I mean, we do detect novel alleles. You can, because we're sequencing single molecules, but where those novel, whether that's germline or cancer cells, I haven't got any experience in that. Yeah.
Okay. And one very last question, quick one, I hope. How many samples can be HLA typed in less than 3 hours?
Okay, so we use that assay only for our deceased donor organ allocation. So we do 1 sample in that 3 hours. So our experience is 1. Yeah.
Okay. Brilliant. As we've now gone over, and I'm sure this is very late for Dianne, I'm sure she now wants to go and relax and probably go to bed. But thank you very much, Dianne. Thank you very much to our first speaker, Professor Dilthey. I hope you have enjoyed today's session. If you are interested in the work of SIP, please go and have a look at our website. We've got some really exciting upcoming events, including at the FE and ASHI meetings and a symposium that will be coming up in October of this year. So please go on our website, have a look and see how you can join in. And with that, I'd like to thank everybody and wish you all a good rest of your day.
Thank you, Naima. Thank you.